I'm SAKAI from University of Tsukuba. Today I'd like to talk about our recent study about understanding the representation of object in the visual cortex. So perhaps many of you here are interested in the representation in artificial neural networks. And many neuroscientists have been working heavily on understanding what's represented in the brain. So let me first explain a little bit about what is the representation, what we are talking about. So when we see an apple, then representation of apple is generated in the brain. And this is not equal to the real apple, because what brain does is the construction of an object in the cortex. So, which is often called the abstraction. And first, let me talk a little bit about what's happening in the brain. So when we see an apple, the pixel-based image is generated on the retina, and then it goes to the visual cortex, first to the V1. And then it goes through the intermediate layers, and then reaches IT, where objects are represented. So in short, the aim of the visual neuroscience to understand how the brain constructs the object in the image. And we think the key to understand this process is in the intermediate layers, especially in the V4. So let me talk a little bit about what's happening in the V4. So in the V4, pixel and the bars are transformed into the object. And we think the key function here is the figure ground segregation. So the V4 need to segregate pixel and bars within the figure and segregate from other object or background. So we think the figure ground is the key function here. And then by pulling the things within this figure, in other words, the property of the object within this region, we can construct the representation of an object. So the point is the figure ground. So today I'd like to talk about this representation in the V4, focusing on the figure ground and contour features. And to approach this problem, we carried out electrophysiology. So we showed a bunch of natural images to monkeys and recorded from the V4 by using a multiple electrode. So I'd like to talk about the three topics today. One is the figure ground, two, contour coding, and three is the correspondence with the CNN. So let's begin with the figure ground. As I mentioned, we showed a bunch of the natural images to the monkeys. So these are some examples. So you may be able to find a whole object in a patch sometimes, and in many cases, only the part of the object can be seen. So this is because we are interested in the V4, the receptive field of the V4, which means that where our cells are looking at is limited, not so huge. So this is about the size V4 neurons are looking at. So first thing we need to do is to get the ground truth for figure ground in these images. So to do that, we carry out the human psychophysics. So we show a bunch of these stimuli to human subject and ask them to judge where is the figure and where is the ground. And perhaps you have noticed that some of these are easy to segregate to figure and ground, but some are very difficult. So there should be some kind of a consistency. So we want to get that data also. So this is one of the example stimulus. So when we show this one, perhaps this is easy for judge. So even we repeat it several times, the subject would answer figure is on the right. So consistency is perfect, so it gets the 1. But if you look at this one, this is difficult to judge figure ground, so it's going to be a chance rate. So this is a perceptual consistency. So let's see the result. So this is the histogram with the perceptual consistency. It ranges between 50-something to 90-something, so it looks okay. We get different kinds of the consistency. And this is some example of these are easy cases and these are hard cases. Perhaps you can imagine these are easy and these are difficult. Okay, so we get ground truth with the consistency. Now we can begin the electrophysiology. So unfortunately, we cannot use a human for the electrophysiology, so we need to exchange human with the monkeys. Then we do the same thing as the human psychophysics, same stimuli and the same procedures. So the point here is that is shown here. So this is one of the example stimuli. In this case, black region is figure, and this red dot shows the center of the CLF of this neuron. So with this case, we get the response of this cell to figure. And then we move this stimulus a little bit to the right. Then we get the response from response to a ground. So in this way, we can get the responses to figure and ground. And we can also change the contrast. In this case, figure was black, but we can change to white. And we are now interested in the figure, so it shouldn't depend on the contrast. And what we expect from this experiment is this. So we should observe, not really should. If the cell likes the figure, we expect to observe the stronger response to figure compared to the ground. So this is what we expect. This is figure ground modulation. And of course, as I mentioned before, even if we change the contrast, we expect the same, not really the same, but a similar modulation. Okay. So we showed these natural patches and silhouette patches because we are interested in the contours, not texture today. So we show the silhouette images too. And here is the ground design of the experiment. So this one is another example of a stimulus. And these many red crosses show the center of the CRF, which means the cells are looking at these locations. So we use the multiple electrode. We can record from some tens of neurons simultaneously. So those are shown in here. And let's focus on this cell shown in a bold cross. So for this cell, we get the response with this cell to figure with this stimulus. And go to the next stimulus. And in this case, figure is black. So we get the response to ground from the same neuron. And with the next stimulus, we get the response to ground, and the next one, we get the response to figure, like this way. So in this way, we can efficiently record the response of many neurons from many stimuli. Good. So let's see the result. So this is an example of the result from a single neuron case. So as we expected, for the response to the figure is greater than the response to ground. And even this is true even if we change the contrast. Looks okay. We observe the figure ground modulation. So let's say another cell. So we still get a difference between figure and ground. And these are just the two example neurons. So how about the population? We recorded from more than 300 neurons, and among them, about a quarter of neurons shows the FG modulation. This is the first time we found, not really, the other way. This is the first report by us to show this kind of the representation in before. So we get the first result. So many neurons show figure ground preference. Now the question is, do they really code figure ground organization across stimuli? So try to answer this question. We try to decode figure ground of a single location from a single neuron. So this shows what we did. So we get a bunch of the stimuli, and we knew the ground truth. And also we get a response from individual neurons. So we can compute how correct these neurons are. We can compute the correct rate of individual neurons. And this shows the correct rate across neurons, mean across neurons. This is a result for natural patches and silhouette patches. So we get correct rate of 55% around that. So it's slightly above the chance rate, but not really good. So we need to conclude that single neurons cannot decode FG organization. So this may be expected because we are now talking about a single neuron. It may be natural that single neuron cannot determine the figure ground of the natural scenes. And of course, we get so many number of the neurons in the brain. So why don't we consider the population? So the next thing we did was the reconstruction of a figure region from multiple neurons. So idea is here. So now we're going to pool the responses of many neurons and then predict the figure ground when some stimulus was presented. But unfortunately, we don't know how the brain integrate or pool these neurons. So we let machine to do this one. So we use a support vector machine to predict the figure ground. So input to the machine is a response of neurons and ground truth. Well, I think I don't need to talk about the support vector machine here in this institute. So let's go to the result. So this is the example of a stimulus. In this case, black is figure, and this is a response, I mean, the activity with the machine. In this case, reddish color shows the figure. So we get the correct responses. And when we change the black to the white, we still get the correct one. And even with the rotation, we get the correct. And this is a pool of the these responses. Actually, the overall correct rate was about 77.77. So it looks okay. Another point is the correlation. As I mentioned before, we have a consistency in perception. Some stimuli are easy to judge, some are not. So these responses are expected to have a correlation with those perceptual consistency. So we computed the neural consistency as a function of the perceptual consistency. And each dot shows each stimulus. And perhaps you can observe the good linearity here. Actually, the number is 0.73. Good, we get the correlation. And another point here is the number of the cells. In the previous, this slide, we pooled 87 neurons. You may think this is large or small, but anyway, we are interested in this number, Y40. Well, I'm sorry. We are interested in why we need, how many numbers of cells are needed to pool. And this shows the correlation between perception and neural activity as a function of the number of cells the pool. And actually, we observed that it gets saturated with around 40 neurons. So the question is why 40? To examine this, we computed the intrinsic dimensionality. Again, I think I don't need to talk about the dimensional tree here. So let's show the result. So what we did basically is just did PCA, principal component analysis. And then in this is a result of the experiment. In this case, we get a significant dimension up to 20. And of course, we can get more dimensions, but these are less than the noise level. So in this case, we say that dimensionality of this system was 20. Okay, but the problem here is that in the experiment, we get a limited number of the neurons and the stimuli. But in natural environment, there are infinite number of the stimuli and so many number of brain. So we need to explorate to stimulus and cells. And after all, we get the dimensionality of 40. So which agrees very well with this result. We needed 40 neurons to judge correct figure ground from natural images. So up to here was the topic of the figure ground things. Next, I'd like to talk about the coding of counter features. Excuse me. So the hypothesis here is the simultaneous coding. So we think individual neurons code multiple features. The previous studies has shown the tuning to bars in V1 and angle in V2 and the convergence in V4. But we think in the V4, neurons need to simultaneously code multiple features because V4 neuron need to code multiple features to represent a single object. Object often consists of the multiple features. And here we focus on the contours. So we computed the actually measure the contour within the natural images. We focus on these four features: closure, curvature, symmetry, and orientation. And among them, closure and symmetry are Gestalt factors, which have been known as an as their importance in the shape perception. And actually, this is the first time that people investigate the selectivity to closure and symmetry in V4. So we want to know the relation between these contour features and neural responses. So to do that, we compute the mutual information between neural responses and contour features. Again, I don't need to talk about the mutual information here in the institute. It's very easy. And let's see the result. Oh, maybe I need to explain a little bit about this. So we get a probability distribution of the contour features within stimuli, and we got the response distribution of the response from cell. So we can compute the mutual information. And this is the result. So these are 4 histograms showing the mutual information for features. Nice. We observe some distribution. So we can say that neural responses are related to contour features. Now the point here is the simultaneous coding. So we expect that amount of the mutual information for these 4 features should have similar magnitude. So let's check that. So this shows the mutual information for each features, color, curvature, symmetry, and orientation for individual neurons. And this is a neuron in the rank order. So we can observe that neural responses were dependent similarly on these features. This is a very nice evidence. So this hypothesis, this supports the hypothesis that the V4 neurons simultaneously code multiple contour features. So let me talk a little bit more about the contour coding. Traditionally, the neuroscientists have examined the tuning curves, which is shown here. Some examples are shown here. So this is the closure value, and this is the histogram of the neural responses. And people often fit this histogram with a Gaussian or cosine function to get the tuning curve. And from this tuning curve, we can observe the optimal feature, for example, for this cell here. So this means that this cell responds more strongly to this closure. And we can get the similar tuning curve for features. So again, we are interested in the simultaneously coding. So we expect to see the significant tuning for the, multiple features. So we computed those numbers. So this shows the histogram of the number of tuned features across neurons. So about more than half of the neurons show the selectivity to at least one of these 4 features, and among them, about half of them shows the tuning to multiple features, in many cases to 2 features. And this is the number, detailed number of the tuned features. So for example, this 24, so it means that the 24 neurons are tuned to both closure and curvature. So let's focus on this curvature and closure. So this is the tuning, cross-tuning to closure and curvature. So in this direction, this is the histogram of the optimal closure. So this is the closure, and this is the number of cells. So in this case, we can say that many neurons are tuned to well-closed contours. Okay, and this is the curvature. So we can say that many neurons are tuned to acutely curved contours with either convex or concave field. And this is a combination of these two. So this is the 2D cross-tuning. So in this case, we observe that the many neurons are tuned to well-closed contour, which is also acutely curved. This may be quite natural in terms of the representation of the corners or shapes. But, well, so this is kind of a peculiar, but one might expect that this reflects the distribution of the stimulus. So we check the stimulus, distribution. So this is the distribution of the stimuli, the same closure and curvature. And in this case, we observe many stimuli included straight lined, because the value of the curvature is close to 0 and the value of closure is close to 5, so it means about straight. So this is a very, actually this response distribution is very different from the stimulus distribution. This is very interesting. So this means that in a sense, information is quite big because the neurons try to detect something barely happen. So we can say that many V4 neurons respond to peculiar and salient features. And we think it may help the representation of the shape because straight line may not represent nicely or efficiently about the shape, but the corners and curvature would. So up to here is the topic about the coding of the counter features. So, I think I talked too much. Let me go briefly about the correspondence with the CNN. So we want to compare the activity of the neurons with activity of the mid layer of the CNN. So to do that, we need some kind of the translator between them. So we use the two-stage convolution network, which is quite simple. And actually, the explained variance of this model is very good, 0.9. So we can use this one as a model. And what we want to examine is a representation within this artificial network. So we are focused on the contours, so let's focus on the contour again. So this is the changing curve I showed before for the neuron, and we compute the same thing from the corresponding model. We get a very similar curve, which is good. And this is another neuron, and how about the population? So this is the population of the curvatures, just as I showed before for neuron, and this is the model response. Actually, the similarity is quite high, 0.96, and this is the Gaussian symmetry. Again, similarity is quite high, and this is two-dimensional cross-tuning. Again, this is for neuron, this is a model. We get a very good similarity. We do have some more examination about the similarity of the response patterns, but let me skip these things. Okay. So today I talked briefly about the representation in V1 before foreground contour features and a little bit about the correspondence of the CNN. And this is the members of our lab who have been working heavily and on our project. Thank you for your attention.
Thank you very much for nice talk.